Those have written up. I have written completely
Yeah.
everything.
Okay. Those you have written. All right. Okay. Just 1 second. Let me. Like there are few questions that I want you to solve. I just wanted to see whether or not the concept is clear for you.
Okay, ma'am.
1 second. Apart from that, up until now, whatever you've studied, you've understood, na?
Yes, ma'am.
Is there any doubt, anything you like feel like ye kya hai?
No, ma'am. No, ma'am.
Okay. Right now, see, I'll be sharing one question paper with you. Okay, you see that question paper and try to just the MCQ part. Okay, just try to do that. If you're able to solve the questions, then it's well and good. If you're not, then it'll like just give me the sense of it. Okay.
Have you shared?
Wait, I'm just sharing my screen. Okay, can you see my screen?
No, ma'am. Not yet.
No, you can't.
No, ma'am. Yes, yes,
Now,
yes.
okay. You also switch on your video. Now, what you do is that these are the questions right in front of you. Take out your paper, like wherever you write, and just write the option A, B, C, D, like that. Okay, wherever you get stuck, do let me know. I just want to see because this chapter is really huge. By the time we reach the end, the ones that you studied in the beginning will be gone. Poof. I don't want that. So just let's just see however many questions you can, and then we'll move on to the topics that we have to discuss.
Yes, ma'am. In a DNA strand, the nucleotides are linked together. In DNA strand, the nucleotides are linked together by a nucleoside differs from nucleotide lacks. Yes, ma'am. Could you go up to the first question again? I guess I read the question wrong. Yes, ma'am. Fourth one. Yes, ma'am. How many questions?
Do it. Don't worry about the questions. Wherever you feel like it's not thought, you leave it. Are you able to solve the questions though? I just wanted to see that it's not a test. I just wanted to see whether or not you're getting stuck in it.
Yes, ma'am. Till now, I'm fine. But then from 6 onwards, let's see. I don't know. Yes, ma'am. Yes, ma'am. Ma'am, 14th one. Yes, ma'am.
Okay, now the rest of it don't have to do because it's from the topics that I haven't taught. So up until now, up until the question number 15, did you face any difficulty?
17.
17.
Ah.
Acha.
Yes, ma'am.
Did you face any difficulty anywhere?
Uh, I don't know.
No, or I
No,
don't know.
no, I don't know. Yeah.
Okay. What is the answer for the first one?
B.
What is the answer for the second one?
Second one, A.
A.
Once again. Nuclear side, right?
Huh?
Sugar is there.
There's only one difference between nucleoside and nucleotide.
Yeah, I
First,
know one thing is missing. It is option C.
so A.
Option C.
Both deoxyribose and ribose sugar belongs to the class of sugar that is
Pentosis.
meaning 5 carbons, right? The fact that a purine always pairs through hydrogen bond with a pyrimidine base in the DNA double helix leads to
The anti-parallel nature.
uniform length all over it.
Uniform length in all DNA.
Because the bond never changes the shape or the length and stuff. It's fixed. The net electric charge on DNA and histones is
Negative and positive respectively.
very. The promoter site and the terminator site for the transcription are located at
Which is 6, right? 6 is option A.
promoter site is located at the 5 prime.
Yeah, from up to down.
Wait, 3 downstream, respectively of the down. 5 upstream, no, 5 prime upstream, and then 3 prime downstream. Because if you remember the transcription unit, we do it like this, isn't it?
Yes.
Which of the following statement is the most appropriate for sickle cell anemia?
Yeah, this one I wrote 7th D.
Correct. This one is such a nice line, no? It confers resistance to acquiring malaria. Do you know why they have written this?
Is my answer correct?
Oh, your answer is right.
I
All
don't
of
know.
your answers up until now are correct.
I didn't know whether it was makes sense with sickle cell anemia and malaria. So I chose option
But
B.
why did it make sense to you? I haven't taught you this.
No. The only thing I know about the sickle cell anemia is molecular disease. So I wrote that.
See, the thing is that because in the case of malaria, the pathogens, they attack the RBCs. If the RBCs shape are like this, the pathogen itself will not get any space to live. That's why a person with
Oh,
sickle cell anemia will not have malaria because malarial pathogen, that is Plasmodium, can't reside. You will be studying this in human health, the next chapter that I'll be teaching.
okay.
Which of the following is true with respect to AUG?
All of the above.
The first genetic material could be
RNA.
with regard to mature mRNA in eukaryotes.
Option B.
Yeah. The human chromosome with the highest.
This one was I didn't understand. But
This
then
one is out of syllabus. The topic is yet to be taught to you.
okay.
Who among the following scientists had no contribution in the development of double helical structure of DNA?
Messelson and Stahl.
Very good. DNA is a polymer of nucleotides which are linked to each other by 3 to 5 prime phosphodiester bond. To prevent polymerization of nucleotides, Which of the following modifications would you choose?
Option C.
Right. Discontinuous synthesis of DNA occurs in one strand. Why? Because
DNA-dependent DNA polymerase catalyzes polymerization and
which of the following step is catalyzed by RNA polymerase?
initiation.
Initiation. Yeah. But yeah. Okay. Which of the following statement is correct about the regulatory protein in the transcription in prokaryotes?
Option C.
Regulatory protein and transcription in prokaryotes. They only increase expression, they only decrease expression. They interact with RNA polymerase but do not affect the expression. So no doubt, right? You flawlessly done the MC. Okay. Now let us move forward and talk about the gene regulation. Now see, in our body or in any organism's body, whenever, imagine it like a big control room, whenever there is a need of anything, let's just say a protein, a particular protein is required, okay? Or a particular hormone.
One second, could you repeat again? I don't know what happened.
I am telling you about gene expression and its regulation. First, I'm teaching you about the gene expression, what it actually means, the word, right? The term. So, see, that is why I'm telling you, like our body, imagine it like a big, big factory. Okay, big factory, big headquarter like that. Now, whenever in our body there is a need for anything, any enzyme to be produced, any, you can say, allergic reaction to happen, any kind of, you can say, procedure to happen in our body, the information is sent at the cellular level. It goes till the cells level. Imagine, cellular level, cell.
Okay.
We have organ systems. Imagine organ system, organ, tissue, cell, like that.
Okay.
So when the information goes to the cell to produce any kind of protein, to produce any kind of hormones, vitamins, any enzyme, the gene expression happens wherein the nucleus sends the information. DNA replication takes place, transcription takes place, translation takes place, then a protein molecule is synthesized. This is what we mean by gene expression. Meaning, whenever there is a need for anything in our body, it begins at the cellular level. It's not very superficial. It's deep, deep, deep down. So, at every level, whether it is DNA replication, whether it is transcription, whether it is translation, at every level, the expression is regulated.
We completed this.
No, we did?
Yeah, only last 3 pages are left.
Lac-Operon is also done.
It's all done.
Human
Only,
genome?
yes, from, you said last 2 or 3 pages are left. I guess from here onwards.
1, 2, 3, 4, 5. Which one? Human genome have I taught you in the previous class? There's a lot. This is a huge chapter. Human genome have I taught you?
No, ma'am. From here I have to start.
Okay. So basically, when people understood about DNA, RNA, everything, now people know that every single organism, including human beings, were made up of thousands and thousands and thousands of genes, right? The total set of genes in the body of any organism is called as a genome. Okay, the total set of available genes. So, in the year 1990, there was this huge project that people started that was called as the HGP, that's the Human Genome Project, wherein people had this thing in mind, this determination that they will map out, they will, like count how many genome or how many genes altogether are there in the human being. Okay, the chromosomes, the genes, the similarities, the dissimilarities, meaning they wanted to go at the molecular level, not even at the cellular level, at the molecular level, to find out the whole entire, you can say, sequence the genome. Sequencing of genome means to know everything about it. Just like how, imagine, DNA if we think about it, is like this, isn't it? We always imagine DNA like this. Now, if we zoom it, we'll see the bases, right? We'll see a sequence that the whole entire DNA is made up of, so on. So this sequencing, they wanted to find out the sequencing of every single thing in our body. Clear?
The thing is, they want to know everything.
Huh?
They wanted to know everything.
Every single gene in every single human. The genetic makeup is the genotype, right? And the reason why we have a certain genotype is obviously because of the DNA, right? Everything we are, everything we offer is because of our DNA. Now, the differences in two individuals will be naturally be reflected, because if we talk about you and talk about me, we look different. We have different voices. We have different, you can say everything, right? But both of us are Homo sapiens, right? We belong to one single species. Despite belonging to a single species, we like normally you are human, I am human. We are very closely related. But despite being very closely related, you have your own set of genome, I have my own set of genome, which makes me me and makes you you. Understood? So that difference that is there in between every individual, they wanted to find out. Okay,
Yes,
that was their aim. So,
but what's the difference between like me and you? What is the difference? Same like DNA. Like, what is the difference between me and you, sir? No.
see, if we talk about me and you or anyone in the world, our genomes are same this much. This much of our genes are same, because you have a nose, I have a nose. You have two eyes, I have two eyes. Right? You have hair, I have hair. Everything like that. But that 0.1% makes you you, makes me me. So this 0.1% difference, they wanted to find out
Only 0.1 difference is there.
in every human being, unless there is some sort of abnormality. Other than that, 0.1. Because we share a common species. Understood?
Yes.
So they can be only known if the entire human genome is mapped. Genome means the whole entire set of genes any organism has. So with the established, now slowly, genetic engineering field came into existence, wherein people were manipulating gene according to their own requirements. That is genetic engineering. Okay.
Manipulating,
Huh?
change, manipulating genes means
Manipulating gene means adding something, subtracting something, like that. Not human, not of humans, but in general of genes.
okay.
So then, because of that, you can like, with the help of genetic engineering, you can extract a DNA, you can cut the DNA, you can multiply that DNA all in the laboratory conditions. Understood?
Yes.
So this was a very big project, ambitious project, and it was launched in the year 1990. Now, what happened? It was an international project, which was a collaborated, like you can say collaboration between many countries. Okay. And the whole entire goal was to map and understand the genes of human beings, right? All genes together of haploid set of chromosomes are known as genomes, right? The total genes that a person has. Now, Human Genome Project, synonymously, it is called as Mega Project. It was a 13-year project. Imagine the duration of this project was 13 years. And it was coordinated by US Department of Energy and the National Institute of Health. You know, there is this website. You might see, like, if you, ever like want to Google something about DNA and stuff, you'll find a website called as nih.nil.ncbi. Okay, like this is a website.
Sorry, I can't hear you again. And
I'm saying that there is this website that is nih.nil.ncbi. Okay, this particular.
NIH,
You just search NCBI. Just search NCBI.
Okay.
When you search NCBI, in that, if you want to find out any sequence of any organism, they have it. They have that information. Whether it is, and it's like so intricate, like if you want to find out what is the difference between your collagen and an insect's collagen, it'll tell you.
Collagen means?
Collagen, the protein that is present in your skin. Collagen.
Okay.
So the difference between yours and insects or any other organism, you can find out. Meaning, the reason I'm telling you is that all the genome possibilities of it's there now. It wasn't there earlier, but because of these works, it is there for us. Now, if you want further to research about it, once you go to your higher studies, you can. So, Welcome Trust UK, they also joined this project as a partner, and contributions also came from Japan, France, Germany, China, and many other countries. So this project finally got completed in the year 2003, not too long ago. So Human Genome Project, it has been called as a mega project. Why? Because first of all, the whole entire, you can say, budget was humongous, right? The huge cost estimated to be 9 billion US dollars, meaning for sequencing of one base pair, they will be requiring 3 US dollars. Then, because you know that human beings, and this is very important, remember that because this is Human Genome Project, you should know what is the size of our genome. If you talk about a haploid set, haploid means one set of chromosome, right? So if you talk about the haploid set, so the number of base pairs are 3 into 10 to the power 9 base pairs. Imagine these many base pairs are present in one set of chromosomes that we have. So we have two. So imagine double the number. So these many had to be identified and they had to be sequenced. Like ATGC, we had to find out what kind of ATGC. So it requires a large number of scientists, technicians, and supporting staff. And then after this was done, the information that they had, obviously it had to be stored, right? They can't take out the information and just let it be. So the storage of data, which required 3,300 books that had 1,000 pages, and each page would have 1,000 typed letters. This is like a huge data retrieval thing, right? However, then they also needed to have the things from the IT sector, right? They wanted to have databases, they wanted to have storage, right? So also one more thing is that there is a big branch of science called as bioinformatics that deals with everything related to that. So this was also developed during this period, which helped the Human Genome Project. So this was the reason why it was called as a mega project. Now let us see what were their aims, what were their goals. So first, they wanted to identify 20,000 to 25,000 genes found in human DNA to determine the sequence of 3 billion chemical base pairs that make the human DNA. Then, after doing all of this, they wanted to store this data in the databases. They wanted to improve it for data analysis, for anyone that is analyzing it. Then they wanted to transfer these technologies to related sectors like in industries, right? Where drugs are made, where injections are synthesized, like that. And also LC, that is to solve any ethical, legal, and social issues like related. Okay. So bioinformatics, that is close association of Human Genome Project with rapid development of a new area in biology. Then sequencing of model organisms. See, apart from humans, now they're like, okay, even though we are doing it for humans, let us do for some other organisms. So we'd have a clear-cut knowledge about different organisms. So they found out, like, see, for example, E. coli. E. coli is a bacteria, but E. coli is not just the only bacteria. There are thousands of bacteria, isn't it? Do you know any other bacteria's name?
Eubacteria.
Eubacteria. Then Eubacteria is like a group.
Eubacteria, then I know many shapes of bacteria.
You might know Streptococcus. You
Oh
might
yeah.
know, you might know this thing, Clostridium. Do you know Clostridium?
No. I have Streptococcus.
Streptococcus, you know. Then Staphylococcus, do you know?
In my 11th book, there are two types of bacteria. One was Eubacteria, the other was
Archaebacteria was also there.
Archaebacteria, yeah.
So there are so many bacteria. So we have chosen E. coli as a representative. Okay. So it's like a ambassador for all bacteria. The reason why we're calling it as the model organism, because this would represent the bacterial community. Saccharomyces cerevisiae is the scientific name of.
Where is it?
Saccharomyces
A
cerevisiae. Here,
free
after
living
recall.
non-pathogenic nematode.
No, good, but use your brain. It's for Coenorhabditis. Saccharomyces cerevisiae.
No, I can't. You're breaking in between. Could you say again?
I'm saying Saccharomyces cerevisiae is the scientific name of.
Where is Saccharomyces in this? I can't find. Oh yeah, it is bacteria. It's one of the bacteria, I guess. No. No, no, no.
It's yeast.
Yeast, yeah.
What is it?
Get it, Saccharomyces. Yeah.
What is yeast?
Yeast, it is used to make beer, wine, beers, and all those.
What is yeast? I am not asking what it is used for. What is yeast?
Anaerobic organism. Yeah, it is in a.
What is it? Like, what kingdom does it belong to? Is it a bacteria? Is it a virus? What is it?
Yeast is bacteria, hopefully.
Only if I could punch you through the screen.
Yeast.
Yeast is a bacteria.
No, it is a virus.
I gave you the option because I knew you will choose it. It's neither.
Oh, yeah.
Neither of the two.
Okay.
What other kingdoms are you leaving behind? Is it a
So-
plant?
Oh, it is a plant.
No.
What is it?
What other kingdoms are you leaving behind?
Monera protesta. That is for plants. Sorry, my bad. No.
It's not a plant, it's not an animal, it's not a bacteria. What other kingdom are you leaving behind?
Just 1 second. I'll ask.
No, no, no. No seconds. Do you know it or not?
What is East?
How many kingdoms are there?
5 kingdoms.
So what are they? Tell me.
Monera, protesta. Oh my god. Sanjitha. Monera, protesta. Then what else is there? Monera, protesta. Once again. Wait again. Monera, protesta. Animals.
Animalia is there.
Animalia. Plante.
What is the fifth one?
One thing I'm missing. Monera, protesta, plante, animalia. What is the third one? And the third one belongs to the east, right?
Yeah. Third one is what the yeast belongs to.
Monera, protesta. Monera, protesta, and one more. 1 second. Just give me 1 second. Monera, protesta.
Then, shall I tell you?
Yeah.
Fungi. Kingdom fungi.
Oh yeah. Fungi. How can I forget that?
You just forget it. How do you do that? I don't know, but you just do. So, Saccharomyces cerevisiae is an yeast. Now, I will not bother asking you which type of it is. It is a unicellular fungi. Okay, it's not multicellular. It's the only fungus that is unicellular. Anyway, I was talking about model organism. So, there are also non-human model organisms to find out their sequence also. So, what are those model organisms? You do not have to remember their base pairs and the genes. You just have to remember their names. So E. coli is there, Saccharomyces cerevisiae is there, then Coenorhabditis elegans is there, Drosophila, Arabidopsis, and Oryza sativa. Last question. Oryza sativa is the scientific name of?
I've heard the name sativum. We learned it within.
Rice. Rhizosativa is the scientific name of rice. Okay.
Nice.
Now see. Let us see the steps. What happened or what steps were used in the Human Genome Project. So they took two methodologies. Okay, one was the EST, the other was sequence annotation. Now, in our genome, there are many genes that are expressed in the form of RNA, meaning transcription till transcription things are done. Translation is done when a protein is required. Okay, so many genes are there that are present as in the form of RNA. So we say that as the EST, expressed sequence tags. Clear?
Yes.
Second thing is sequence annotation. So sequence annotation is basically the whole entire concept of sequencing the genome that has the coding sequences also that have, because as genome has coding, non-coding, like these all the sequences are present all together. There are many genes that do not code for anything, but they are still there. Okay, so sequencing of all of that is done. Now, how do the sequencing is done? So first, the organism's DNA is taken out. Right, DNA could be taken out from anything. It's present in the saliva. It's present in the hair strand. It's present everywhere. So the DNA is extracted. Then that extracted DNA is cut into smaller pieces. Now, obviously, since a DNA is something that you can't see normally, right? So the thing that you use to cut it with can never be scissors or knives, but it would be molecular scissors. Okay, these are enzymes that cut the DNA. Okay, so now you've cut the DNA into small pieces. Now you will increase the number of pieces by making clones of it. Okay, you'll amplify those DNA so that you'll have a lot of things together. Then you will take automated DNA sequencers which will do the work for you because sequencing machines were made by that time. Okay, so then it was sequenced. Clear? So there are 4 steps: taking the DNA, cutting into fragments, cloning it, and then sequencing. Okay. Now the commonly used host at that time, they were bacteria and the yeast, and the vectors that were used to deliver the piece of gene into the host. And see, why we are delivering it? Because we are cloning it. So cloning is done in a vector. For example, do you know how many times does it take for a bacteria to divide?
How many times?
Like how much time? Sorry, how much time does it take for a bacteria to divide?
1 hour.
20 minutes. Bacteria divides in every 20 minutes. Okay. So you take the gene of interest, you put it into the bacteria. When the bacteria will divide, the gene of interest will divide alongside. So you'll have multiple copies and within no time. So that is cloning. But for that, you have to transfer the gene into the bacteria. So here the bacteria becomes the host. And the thing that you are using to transfer it are called as the vectors. So here the vectors used were bacterial artificial chromosome and yeast artificial chromosome. These are the names of vectors. Clear?
The DNA is put into the bacteria. So the DNA is put into the bacteria.
For multiplication.
Okay.
Okay. So now after that, the fragments, they were sequenced using the automated DNA sequences, which was working on a principle developed by Frederick Sanger. Okay. So he is the one that actually made DNA fingerprint. Okay. Also, he developed the method of determination of amino acid sequence in proteins. These sequences were then arranged based on some overlapping regions. Okay, all of these things were done, and lastly, it was like aligned and sequenced and arranged accordingly. And the sequence of chromosome 1 was completed. Like the first chromosome to ever be sequenced was chromosome number 1, which was completed in 2006. No, actually, it was the last chromosome, not the first one. Chromosome 1 was the last chromosome to be sequenced. out of all the 24 human chromosomes that are there. Clear?
Okay.
So this was the methodology. Now the features. Let's just see what they found out. So the human genome contains 3164.7 million nucleotide bases. Imagine that many million nucleotide bases are there. The average gene consists of 3000 bases, but the size varies greatly. The largest human gene is dystrophin that has 2.4 million bases, and TDF gene is the smallest gene with only 14 bases. Now, the total number of genes that is estimated is 30,000. That is much lower than what we estimated, that is 82, 1,40,000. It is not that much. And almost 99.9% nucleotide basis is same in all people. It's that 0.1% that makes it different. The functions are known for over 50% of discovered genes. The rest, we don't know the functions. Less than 2% of the genome are responsible for coding of the proteins. Repeated sequences make up very large portion of human genome. So in human genome, a lot of sequences are repeated many times. So that is why the repeats are there. Okay, the repetitive sequence are stretches of DNA that are repeated many times, sometimes hundreds to thousands of times. They have no direct coding functions, but we see that they are important because that's how they are. Okay, that's how we are made. Chromosome 1 has the most number of genes, that is 2968 genes, and the Y has the fewest number of genes, that is 231 genes. Now, scientists have identified 1.4 million locations where single base DNA differences occur in DNA. We call that as single nucleotide polymorphism. Okay, single base differences. One base changing at different locations is what we call as SNPs, which is known as SNPs. Clear? This much is clear? So these were the features, these were the findings. Now comes the application and future challenges that they could have faced. The completion of first phase of human genome, it was, it is compared to the discovery of antibiotics, because now if you know about the genome, if you know about where the mutation could occur, where is the problem, then a lot of diseases could be treated, isn't it? A lot of chromosomal disorders could be treated. There are more than 1200 genes that cause common cardiovascular ailments like the heart-related problem, endocrine diseases like diabetes, neural diseases like Alzheimer's, cancers, and neurological ailments. So we can remove the disorders from happening if we know the genome and how it works. Right? Defects could be treated, and it will be possible to study interaction between various genes, proteins, as well as mechanisms of forming tissues, organs, tumors, or switch to different developmental stages with the help of this huge information that we have received. So it holds promise of healthier and longer living, designer drugs, genetically modified diet according to the need of individual humans. Right? That is like, it makes it specific. This much is clear?
Yes.
Now we come on to DNA fingerprinting, the next topic. Just like how there are fingerprints that are unique to every individual,
Yes.
right? I cannot unlock your phone, you cannot unlock my phone. Same way, DNA fingerprinting is also there, which is like the fingerprints of DNA. That tells us how you and I are different. Now we want to know. Okay, so this DNA fingerprinting, it is a technique to find out the nucleotide sequence of certain areas of DNA which are unique to each individual. But there is an exception. Monozygotic twins are 100% similar. They're not different at all. You can, like, every single individual on the planet Earth are different from one another because of that 0.1%, except for monozygotic twins. Now see, there are two types of twins, right? Identical and non-identical twins. Do you know how they are made?
When there are two eggs formed, then there may be a possibility where there's a boy and a girl. But in one egg, and it separates the zygote if it separates. In like from the one egg itself. One, yeah. that forms identical twins. Like both are female, both are male.
Very good. Because of like the splitting of zygote, monozygotic twins are made that are identical, and non-identical for the different reason that you said, that maybe two gametes fuse or like that. So 99.9% base sequences among humans is same. It's that 0.1% genome or 10 to 10 to the power 6 base differences in the base sequence, right? The differences occur not only in genes, but they also occur in the repetitive DNA. So DNA fingerprinting, it involves identifying differences in some specific regions in DNA we call as repetitive DNA. We wanted to know, like we see how the repetitive DNA are different from each other. Okay, so it's like different from the bulk genomic DNA, and the bulk genomic DNA forms a major peak, and the smaller ones are called as the satellite DNA, the ones that we are concerned with. Okay, so the satellite DNA, they have like different composition. Like they could be rich in adenine thymine, like they'll have more adenine thymine groups, or they could be rich in guanine and cytosine more. The length of the segment and the number of repetitive units, these are things make them different from the genome that is present in the normal bulk DNA. Then there are two things, the two categories of the satellite DNA. There's VNTR, that is variable number of tandem repeats, or you can call it as mini satellite. Okay, it is basically a small DNA sequence is arranged tandemly in many copy numbers. Copy number means a lot of repetitive forms. Okay, and this shows a, you can say it's different from individual to individual. Second is the SSR, single sequence repeats. That is microsatellite. It's even smaller than that. Again, repeats are there, unique to every individual. Clear?
Yes.
Now, these sequences, they normally do not code for protein, but they form a large portion of human genome. These sequences, they show high degree of polymorphism, and they form the basis of DNA fingerprint. Now, polymorphism is the basis of DNA fingerprinting. Okay, the way the genes are present at every level. Now, DNA is present in every tissue, every cell, such as blood, hair follicle, skin, bone, saliva, sperm, anything. You name it, DNA is present in there. So, like for forensic application also, like in a crime scene or paternity disputes like that, the uh, the specimen could be taken for testing, right? And find out to which person it belongs to, or to which person it is matching. So as polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, so you should know what is the meaning of polymorphism. Like I told you, it's the difference, it's the variation that is at genetic level. And this is happening because of mutation. Okay. So allelic sequence, like frequencies or the sequence that are there, that are normal repeats that are there in the genome of the human being, that is also described as many a times DNA polymorphism. Clear?
Okay.
Now remember, when we talk about this, we talk about the coding sequences, the non-coding sequences, and everything. So the basis of DNA fingerprinting, along with the polymorphism, is your VNTR, that is variable number of tandem repeats. Again, the complete repeats that are there. The technique of DNA fingerprinting was developed by Alex Jeffries. And now again, let us see what happens here. Isolation of DNA, see the picture. The DNA is isolated, okay, from the blood sample. Then the DNA is cut into pieces with the help of restriction enzyme. And then the separation of those fragmented DNA is done by gel electrophoresis. Okay, the strands are separated. Then the blotting technique is done, like whatever strands have been separated, just like how blotting sheets are there for absorbing oil from the face, same way blotting sheets are there to take up the DNA from the membrane. Okay, and then after that they are radioactively labeled, and then they are like X-ray crystallography is done to study the pattern. And then the pattern of two individuals are matched to see the similarities or the difference. In the case of dispute or in the case of this thing, crime scenes. Okay. This is your basic step of DNA fingerprinting. Now let us see the practical applications. Paternity-maternity disputes to find out who the child belongs to. Second, criminal identification and in the
What
case.
is paternity-maternity disputes?
Huh?
What is paternity-maternity disputes? The parents' child, right?
Paternity. Huh?
It is the like I don't understand paternity-maternity disputes.
Oh, paternity-maternity disputes is that wherein like a couple or like a partner has accused the other partner that this child is not theirs. So in that case, they could have had the the gene and DNA, GNA. So that would be sequenced to find out who the child belongs to or is similar. Clear?
Yeah, but DNA fingerprinting is done for that.
It could be done, yes. Understood. That will give you. So personal identification also, like you can understand about your genes, which gene of yours has any kind of mutation, that you can also do. And close relations in an intending immigrant. So there's an immigrant. If you want to know whether you're not related to that person, again, go have your DNA fingerprint. So done? Clear?
Yes, ma'am.
We're through with the chapter, Sanjitha. We're done. We've done it.
Oh,
The chapter
finally.
is done. Now, what you're going to do is take screenshots. Do you take the screenshots of these parts?
Ma'am, could you post this for screenshot? It'll be not really clear.
Do you want me to give you the notes?
Yes.
Yeah, see, I could give you the notes, but the thing is that I don't know whether or not, because this is a very long PDF, so it goes wonky. Okay, let me see. I'll send it to you. And if you can't see, then let me know. I'd have to go for a difficult route. Hmm. You do me molecular basis. I'm so glad this chapter is. Which chapter are we going to begin next?
What is the next chapter, ma'am?
I'll start human health and diseases. Okay. I can't handle one more very long, boring chapter. We'll do one very interesting chapter, then we'll move on to evolution. Is there any
Yes.
particular chapter? Like you have your summer vacations going on?
Yes, ma'am.
Till when?
29th my school starts.
This 29. And how many chapters have been done in your school before the school closed?
Till this chapter.
Till this, how many chapters have I taught you?
2 chapters.
Principal and this.
Yes.
And when is your exam?
September mid.
What is the portion?
80%?
Huh?
Yeah.
80%?
Yes. I don't know, 85%, I guess. 80%, 80%.
We'll start with human health and disease. Okay.
Okay.
This is a very nice, easy peasy, very breezy chapter, very pretty chapter. I find this chapter very pretty. It's very nice. It's fun on the eyes, it's fun on the ears, it's not difficult at all. It's like you just have to listen to it like a story. And but don't underestimate it. So, human health and disease. So, first, let us see the, what do you call it? The introductory things. So, first of all, when we are associated with these things, we should know some pioneering people's names, just like how you know the names of Mendel and Erwin Chargaff and Watson and Crick. So the father of medicine, Hippocrates. Hippocrates is known as the father of medicine. Then William Harvey, he discovered the blood circulation. Now talking about what actually is health. Health is a state wherein a person is fit in terms of physical, mental, social, and psychological well, all that. If a person tick marks all the things, a person is considered healthy. If any one thing is compromised, the person is not healthy. Now, our health could be affected by numerous reasons, just like genetic orders, right? The things that we are born with, infections, the things that we acquire while we are living after we are born, and also because of the lifestyle that we choose, right? Because there's genetic disorders we don't have any control over. Infections, let's just say we don't have any control over that also. But lifestyle is totally in our hands, which also impacts our health, right? A person that has a lot of complex carbohydrates, a lot of processed and oily and stuff, the person is like on the verge of getting a lot of, you can say, high blood cholesterol, heart related disorder, diabetes and stuff, right? So it's the lifestyle that also affects the health. Now, what is the thing important to maintain a good health? Obviously, a balanced diet, never skipping a diet that contains proteins, carbohydrates, all the necessary things, right? Roughage, what vitamins, minerals, good fats, water. Personal hygiene, that is also very important, and regular exercise. These three things are extremely important to maintain a good health. Do you exercise?
Yes.
Now
I'm
talking about, huh? You're into
I'm
sport?
yes,
What
badminton.
sport do you play?
Badminton.
Oh, wow, nice.
I'm the sports captain of the school.
Very good. Something that made me very happy. Very nice. All right. Talking about diseases, what diseases are? So when in our body, any organ or any organ system gets affected, we say that we have a disease. Now this effect on the organ or the body part could be because of anything, right? That is what we call as a disease. Now, the diseases, they are of two types: congenital and acquired. Congenital is the one that a person is born with. Something that a person did not have in the world, instead had as like while he or she was born. And the second type is the acquired disease. The disease that a person develops after the birth, right? In the young age, in the old age, in the mid age, anyway. Now, these acquired diseases are of two types: infectious and non-infectious. Infectious are also known as communicable diseases. Non-infectious are also known as non-communicable diseases. Give me one example of infectious disease.
Fever.
Fever is infectious disease.
It can transfer, right?
No.
Yes. Cold, fever.
Cold can. Fever can't.
Fever can't. If one family gets fever, the whole family gets fever.
It's the cold. It's the cold that a person gets that gets transferred. Fever is a symptom. Fever is not a disease. Cold is a disease.
Okay.
Fever is your body fighting with that. That's why we
Okay.
get a fever. Whenever our body is fighting with any pathogen or any foreign particle, and the body's temperature rises during that. That is what we mean by fever. Okay.
Okay.
Non-infectious diseases, one example.
Cancer.
Very good. Best example. Cancer. No matter if you sit on that person, hug that person, the cancer can never be contracted. Now, those agents that cause diseases are called as pathogens, right? So the disease causing microorganisms, not micro, disease causing organisms are called as pathogens. Now you have bacteria, that's a pathogen, virus, fungus, protozoan, and helminths. You have 5 pathogens that are mentioned, each of which related diseases you're going to study. Okay. So now, how does a pathogen cause diseases? The first thing that a pathogen has to do is to enter inside us, isn't it? So it can enter through the nose, it can enter through the mouth, it can enter through the cut areas, right? In all of that cases, it could enter, it could multiply and interfere with the normal activities that we have going on. And you should know that the pathogens, they're so clever that they have this property that they can adapt to the environment of the host body. They can adapt. They sometimes have this modification in them. Like for example, if there is any pathogen that is localizing in the intestine area, okay, it is infecting the intestine. So you know that in our intestine, complete digestion happens and absorption of food also happens. So these pathogens, why are they there? They are not there to cause disease. They are there for their own survival. They utilize you for their own survival. So, like if they are present in the small intestine, you know that small intestine has already the digested in food there. So they get ready-made digested food For that, they don't have a digestive system. This is a modification. Understood?
Yes.
In our stomach, there are many pathogens that affect our stomach. Our stomach has hydrochloric acid in it, which has a very low pH. It's a very strong acid. So they have mechanisms to protect themselves. Understood? So
What,
that's
what happens if we drink hydrochloric acid?
what happens.
If we drink hydrochloric acid?
Will I? It will
No, that's
burn
not.
everything.
Okay, okay.
Hydrochloric acid is present in our stomach. It is there, yes, indeed. But our stomach lining is also protected from it by mucus.
Okay.
Understood? And one more thing, you might have noticed that when you are fasting or when you're very hungry and you haven't eaten for a while, the stomach starts aching.
Yes.
Why? The hydrochloric acid starts acting on the walls of the stomach.
But it is already covered, right?
No,
No.
mucous membrane is there, na? So it
Yeah.
starts corroding that.
Oh, okay, 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 okay.
Okay. Now, the first set of diseases that we have to study are bacterial diseases. Okay. So, back for studying the bacterial diseases, I have written the name of the disease, the causative organism. All of this you have to remember.
Oh, by the way, which chapter are we doing right now?
Huh?
Which chapter are we doing? The name?
Human health and disease.
Okay, okay.
I'm coming almost to the quarter of the chapter. And which chapter, ma'am, we are doing?
No, I just want to know the chapter name.
Human health and disease. Okay. So now we're going to study the various bacterial diseases. So there's typhoid caused by Salmonella typhi. Okay, all see, even though NCRT doesn't have every single bacteria name given, but you have to remember it because I will give in the test. Okay. So pneumonia caused by Streptococcus pneumoniae, plague caused by Yersinia pestis, tetanus caused by Clostridium tetani, leprosy caused by Mycobacterium leprae, cholera caused by Vibrio cholerae. Then there's diphtheria and dysentery caused by Corynebacterium diphtheriae, and tuberculosis lastly caused by Mycobacterium tuberculosis. Clear? Now you can see that here in the organism's name, I have underlined the name separately, right? One is the genus name, one is the species name. So remember this as a thumb rule that whenever you're writing a scientific name, you have to underline the two names separately. The genus will always begin with a capital letter. The species will always begin with a lowercase letter. And when you're underlining it, you will underline it separately. This is a very important thing you have to remember because I cut marks on that and many teachers also cut marks on that. Okay, so this is like a nomenclature rule that everyone has to follow. Clear?
Yes, ma'am.
So now the question comes along, how do you remember these many diseases? So I have a mnemonic for you. The mnemonic is, you remember the mnemonic and then the rest will come. The mnemonic is TPPT LCDT. TPPT LCDT. T stands for typhoid. Okay. P stands for pneumonia. P stands for plague. Okay. P stands for plague. Then you have another T. You only tell me what will it stand for?
Typhoid.
Typhoid already done. Again, typhoid.
Tuberculosis.
Tuberculosis. Very good. L stands for
Tetanus. No, another T for tetanus. Another T for tetanus. The down one.
yeah, yes. Very good. Then L stands for leprosy. Okay, it's okay. I don't expect you to learn it right here only. So leprosy. C stands for cholera, right? And D stands for diphtheria and dysentery.
One second. TPPT LCDT.
TPPT LCD. Remember this mnemonic? All the diseases will bear. The mnemonic for viral diseases is my favorite. You will love it. This much is clear?
Yes.
Any doubt?
No, ma'am.
Okay, now you know down.
Please, no. Pray.
Yeah, now this is a different chapter. It's a nice chapter. You don't have a lot of things to note down. You take out a fresh page. You write down.
Ma'am, I will continue. Only few minutes are left. Please.
Leave pages for it.
I'll leave it. Please. My hand
But
is paining.
I need to rest. Your hand is paining from what? Listening.
No.
Right. Take out a fresh page, write it. Now this is a fresh chapter. You need to like learn nicely this one. It's very intricate also. Very nice. Very nice chapter. No doubt.
Yes, ma'am. Yes, ma'am. Pana. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am.
Any doubts?
No, ma'am.
Why do you sing? No, ma'am. Okay. So let's see. NCRT has discussed few diseases in depth, not all of them, few of them. Okay. So the one that gets, like a lot of question gets asked from is typhoid. So typhoid is caused by the bacteria Salmonella typhi. Another name for typhoid is enteric fever. So if they ever in MCQs give you enteric fever and it's causative organism, don't get confused, it's typhoid. Now, every pathogen has some place in our body which it loves to reside in. Not everything, but some. So, the site of infection in the case of typhoid is small intestine. Okay, these bacteria, they harbor the small intestine. So, how do they enter? They enter through the contaminated food and contaminated water. Talking about the symptoms, so there's sustained high fever, that is, like 39 degree to 40 degrees Celsius, which is higher than obviously the body temperature. Weakness, stomach pain, constipation, headache, loss of appetite. These are few of the symptoms of typhoid. Now, if this disease is not treated, it could lead to complications as well. That is intestinal perforation. Perforation, you understand? Creation of pores in the intestine, tiny holes. Imagine intestine would have holes in it, and also the death could happen in severe cases.
Wow.
Wow, the person will die. Now see,
Whole time.
yeah, holes in the intestine. These bacteria can do that. Also, you remember like when the COVID time was there was a test done to confirm whether a person is COVID positive or not. What was the name of the test?
They use a earbud and this. Yeah,
Name of that. I am not asking you the process. Name of the test.
COVID test.
No. It is COVID test, but what was the name of the test?
There's a test. There's a name of the test also.
RT-PCR. Remember RT-PCR? No.
RT-PCR.
Hmm. So same way, if you would have said, I plan out the whole entire lessons. Okay, I like plan out what I'm going to say to the child, and the child is going to reply, and then I'm going to do a thing, and the topic will. I ask you questions, you don't answer them. The whole entire thing that I planned is gone. Now see, if you would have said RT-PCR, so I would have said same way. There are different tests for different diseases. The name of the test used for typhoid is Vidal test. But since you did not say anything and my whole entire planning failed because of you, so you should know the test, confirmatory test of typhoid is Vidal test. Clear?
Okay.
Now see, there is this one phenomena that is associated with typhoid, and that is Typhoid Mary. A person was named Typhoid Mary. She was a cook by profession. Her name was Mary Millon. What she did is that she spread typhoid through the food that she was cooking. Like the people that had the food cooked by her got typhoid. So that is why she was named Typhoid Mary. Clear?
Also,
Any
yeah.
doubts?
How can you do that?
Huh?
How can we do that?
How can you do what?
Food where you get type like how did she make like that?
She contaminated the food. She like maybe I don't know to imagine, but she contaminated the food in some way or the other. She was using not good water, not good like properly washing the fruits and vegetables like that. Then the pathogen would have persisted, and she caused typhoid to all those people that had the food cooked by. Clear?
Yes.
So we'll keep it till here only for today's class. Now in the next class, we'll begin with the rest of the things, and we'll do the viral diseases and everything in the next class. Up until now, any doubts?
No, ma'am.
Okay, so I'll be sending you the notes of this particular chapter, the molecular basis. Complete your notes and start to learn that chapter. Okay?
Yes, ma'am.
All right. Okay, then I'll see you in the next class.
Thank you, ma'am.
Most welcome. Go and study now. Bye bye.